Welcome everyone to the AATN seminar today. Our speaker today is Živa Urbančič from Durham University, who will be talking on clustering scRNA-seq data. The floor is yours.
Thank you. And first of all, I would like to thank the organizers for inviting me to give a presentation. As Bastian said, today I would like to tell you about single-cell RNA sequencing data and also on how to cluster it. And I would like to split my talk into four main parts. So for starters, some biological introduction is necessary. So we will be briefly talking about the cell and the cellular processes. We will then discuss what single-cell RNA sequence data is, discuss some of its peculiarities, how it's usually dealt with. And in the second part of the talk, we will see a practical example, so the specific data that I've been working with. So this is the Thirsty Fly dataset, and I will present some initial attempts at clustering it. You will see that I struggled, and then in the end, I would like to present a method that seemed to perform well. And it is topologically motivated, so I think it fits nicely with the seminar. Right, so let's start with the biology. So we will be looking at cells, and each of the organisms has a unique kind of instruction set for anything that your body on the level of the cells wants to do. So this basically recipe is the same for each cell, and it is the DNA. DNA is a double-stranded molecule that is built out of nucleotide bases. And we know there are 4 that always come in pairs. So we have cytosine, guanine, adenine, and thymine. And when I say that they come in pairs, if we have, let's say, cytosine on top, so on the top strand, we have guanine on the bottom. And these encode instructions on how to do things. But this is not the only genetic information within each cell. So whenever a cell wants to do something, it needs to get the instruction from the nucleus where the DNA is to whichever organelle performs the task. So how do they do this? The process of doing this is called transcription. And basically what happens is whichever genes we want to copy, we temporarily separate, the cells do this themselves. The DNA is temporarily separated. And basically we make a negative of one of the strands by basically combining the correct pairs of the bases on the bottom. And when this is done assembling, it is able to detach, and it's its own molecule called the messenger RNA molecule. And this molecule is then able to leave the nucleus and travel to whichever organelle it needs to. And messenger RNA molecules, the amounts of them in the cell vary all the time. And this is based on whatever the cell is trying to do, or whatever the stimuli from the environment is and how it is responding to it. So this is actually a very interesting molecule to look at if you want to know what the cell is doing at each given time. And this is basically what we'll be looking at today. So how do we obtain data summarizing these mRNA molecules? I will not surprise you by saying that we start with a sample, some tissue, and in the first step, what we need to do is separate each cell from all the other cells and isolate it in, that is usually the oil-based emulsion, and it is called a droplet. So we put one cell into each droplet. From this droplet, then mRNA needs to be captured. And because mRNA is a one-stranded molecule, it is not as stable. So the next step is to stabilize it through a process called reverse transcription, where we basically make its match with the bases on another strand. And this resulting molecule, the double-stranded molecule, is called the copy DNA. Right, so once we have that, what we need to do is go from a molecule to something that we can work with on the computer. And basically what happens in this step called sequencing is you take each base and you record it in a sequence for a computer to analyze. And we also follow this with a step of quantification, where we basically just look through the sequences that we obtained and identify how many times each gene was copied. So to summarize, we have a cell, and for each gene that we want to measure, we count how many times it was copied into mRNA molecules. Because we're able to do this for tens of thousands of genes, what we obtain is a long vector for each cell, and there can be from hundreds to tens of thousands of cells in our data set. So what we get is a big, big point cloud that we need to analyze. So what do we want to do with this type of data? The fundamental question that we're trying to answer is, what does each gene do? And here we can understand this question in many ways. So maybe two sub-questions that we might be trying to answer in our analysis is, how does it respond to stimuli from environment? So basically, how does gene expression change as we change the conditions? And maybe another view that we might take is to ask, what role does a particular gene play in the function of the cell? And here, I would like to say that these two questions often interact with each other. So for example, if you're studying the response to stimuli from environment, you might not want to look at an individual cell, but instead you want to look at a cell type, which is where the second question comes into play. And you also see how clustering might be important for analyzing these types of data set. All right. So this is a very specific type of data that you might work with that has its own peculiarities. And these can be due to how the data is obtained or just the biological variance that is present in it. So some things that we can notice in the data, I will list them here. So the first one is batch effect. Oftentimes you need to split your sequencing into batches just because it is so labor and time intensive. And each batch might differ from all the others based on the specific conditions it was done in. So maybe different batches were performed by a different researcher or at a different time of day, or even you might have used a different slot in the sequencing machine. So all of these might play a role in what the results are like. Second thing, we need to make sure that the data that we use is good. And here, I mean the droplets that we obtain the mRNA from need to contain healthy cells and exactly one cell per each droplet. And this is something that we can deal with in the pre-processing step, and actually we are quite good at dealing with this. Now, the next thing that is very peculiar and is very specific to single cell RNA data is the dropout effect. And this is basically, you can understand it as false negatives, false non-expressions. So how do you see that in your data? Let's say that you have two genes that are correlated or co-expressed, meaning that when one is expressed, the other one is too, and when the one isn't, the other one is not either. So if you plot for each point on the x-axis gene 1 and on the y-axis gene 2, you get a point cloud, kind of like this one here. And what you would expect is to have a high density of points along the diagonal or some other increasing line. But if dropout is present, what will actually happen is that the regions close to the axis, like here and here, will be quite densely populated. And this happens because one gene is expressed and the other one is not. Another thing that you might notice is over-dispersion in the anti-diagonal direction. Yeah, so this might be something that you want to take care of before you do any analysis. Yeah, there is also some natural variance always present in the expression data. And this is due to many factors, but one thing that I already mentioned is that the amounts of mRNA vary all the time, depending on many different things. So natural variance will be present just due to that. And lastly, these experiments, the sequencing experiments, are already very expensive. Now imagine having to, for each cell, being able to already tell what its type is by just observing it, or how it's responding to your experiment. So this would be almost impossible in almost all cases, which is why ground truths usually don't exist in this world. And also benchmarks are not agreed upon. So maybe some more mathematical properties of these type of data sets. So the first thing you need to ask yourself is, when is a gene even expressed? So I don't know if I mentioned, but the entries to these data sets are integers and positive integers. So basically they're ranging from 0 to some, I don't know how large value, but it can be thousands. So where is the threshold of expression versus non-expression? So actually the threshold is around 0. So when you see a 0, you usually think, okay, this gene is not expressed in this cell. But here you need to be a bit skeptical because of the dropout effect. When you see a 1, you might also be skeptical. You are not convinced that this one is not just some artifact of something else. But usually you trust the entries from 2 onwards. But then the question is, 0 and 2 are quite close in most metrics that we look at, versus 2 and 1,000, which are usually quite far. But in this case, 2 and 1,000 are both expressed, versus 0 and 2, where one is not expressed and the other one is. So you might want to binarize your data, but another thing is that we actually care about the scale of the expression. So how do we mitigate all that? This is basically up to you to decide how to do this. Second thing you might ask me is, how is this data distributed? And if you make any assumption, it is that it is either negative binomial or Poisson distributed. So here on the right, you have 2 plots. These are histograms of gene expressions for 2 different genes. And I fitted both PDFs for both the negative binomial and for Poisson distribution to them. And you can see that on the top, the negative binomial is quite a good fit, where on the bottom, neither of them is a good fit, but a mixture of them might be. So yeah, whatever assumption you make will basically, it will not be okay on all of your data. And here I would like to point out that this high value at 0 might be due to dropout, but it also might be just due to having a bimodularity in your data set. A third thing that you might want to consider when you're doing this type of analysis is that genes that are not important for the specific task often outweigh the ones that are. And this is a pure numbers game. The amount of genes that are usually important for these types of analysis are in tens, so up to 10, let's say. whereas the ones that are not important are all the others in a data set of 10,000 genes. So just by the amount of genes that are not important, they will kind of outweigh the ones that are. Here I will not say too much, but this data also suffers from the curse of dimensionality, which might mean that you might need to adjust the methods that you're using, do some dimensionality reduction steps at times, or even do experiments in batches. And lastly, Euclidean distance tends to prefer scale over pattern, and I will show you what I mean by that later on. Whereas the pattern of expression is better measured by cosine similarity, so you might think of adding this to your analysis. So cosine similarity is if you take two vectors, the cosine similarity between them is basically the cosine of the angle between them. Or in other words, you can take the dot product and then correct it for the scale. So to illustrate my point before, we have three vectors here. A and B are very similar in scale, but the pattern of the expression is kind of exactly opposite, with the exception of the last one, versus C, which is the same in pattern of expression with A, but the last entry is much smaller, so it's much smaller in scale. So as you can see here, A and B with the Euclidean distance are considered closer than A and C, whereas the cosine similarity picks up that A and C are much closer in pattern. So this is something that you might want to include in your analysis. Okay, so how do bioscientists typically work with this data? So the first necessary step is pre-processing, where you try to deal with many of the things that I listed before. Second step then is feature selection. And this is to deal with what I said before, that genes that are not important for your analysis might outweigh the ones that are. So in this step, what you're trying to do is throw out any genes that you can identify in advance that they are not interesting on your data set. And you might do this by basic statistics like mean and variance estimation. And here you might follow different reasonings for why to include certain cells and not others. So this is up to you. The third step is then clustering. And sometimes, as I said, because of dimensionality issues, you might do a step of dimensionality reduction before. But once you attempt clustering, most often the bioscientists use community detection methods, which are based on k-nearest neighbor graphs. And one thing I would like to point out here is that these methods tend to perform really poorly when the amount of data that you have in terms of the amount of points is small. And lastly, basically is a step to obtain insight from your clustering. Basically, what you want to do is detect the genes that drive your cluster separation. So you want to identify genes that are high on one cluster and low on others. All right, so let's go on to the second part of my talk, where we talk about an actual example of a data set. Here I would like to thank my collaborator, Vincent Corsey, for making it available to me. He was part of a group of scientists studying the response of fruit flies' brain, so the neurons, to water deprivation. So they had 4 groups of flies. One was the control group, one was water deprived for 6 hours, one was water deprived for 12 hours, and one was first water deprived for 12 hours and then sated. They obtained gene expression data for neurons in the fruit flies involved in the experiment. And in the first step of their analysis, they tried to kind of group the cells by cell type. So here are the main cell types. And they were particularly interested in the monoaminergic one, which is the small one here in mustard yellow. Yeah, it's the smallest one. It has 855 points. And what they wanted to do is cluster it further. And as I said, their clustering methods tend to not perform well on small data sets. So they struggled and they asked me for help. So my task was to find 4 subclasses of monoaminergic neurons. And at this point, I might also explain what monoaminergic neurons are. So neurons transmit signal from one neuron to another through a synapse. And here, this transferring is done by transmitter molecules. And when those are monoamines, the neuron is called monoaminergic neuron. And there are 4 main types in fruit flies. Namely, the monoaminergic neuron might be specialized for dopamine, serotonin, octopamine, or tyramine. And those are the 4 classes that I'm trying to find. The second task was to then, if I performed the first task successfully, to then cluster dopaminergic neurons further. And the data set that I got was 855 times 10,000 matrix, where each row was a neuron and each column was a gene. Right, so let's go to the first task, where I'm trying to cluster this into 4 classes. Here, Vincent was actually able to give me quite a lot of information. Namely, he was able to give me 7 important genes that kind of drive the specialization of a neuron for one of these 4 types. And I listed them above. And not only that, he was able to give me combinations that are associated with one or the other type. So, for example, he told me that if PLE and DAT are highly expressed, I can be certain that this is dopamine. So using these relations that were given to me, I was able to go through the data set and identify certain points for which I was certain which type they are. And I called those representatives. So what you see here on the plot is the matrix that I had, but only on the 7 columns belonging to the important genes, projected down to 2 dimensions with UMAP so that I could visualize it. And then I annotated the representatives of each class. And what I'm happy to see in this class is that kind of the location of each point is kind of indicative of which class it belongs to. Right, so the first thing I tried to do was I tried to use the standard methods for clustering. So here, it's actually 4 plots. 2 are on this slide and 2 are on the next. So here I would like to convince you that it is difficult to do clustering successfully with the existing methods. Something that, for example, bothers me with k-means is that I have this dark blue cluster here that has 3 different representatives in it, which you might say I can do another step of clustering, but I would like to use this for completely unsupervised tasks where I don't even have the representatives. So how would I know to then do another step of clustering and not just be satisfied with this? So this is k-means plot, this is the plot, this is db-scan plot. This one is spectral clustering, where here I actually used the cosine distance. And this one performs the best, I would say. And the last one that I tried was Lovan algorithm, which is one of the community-based algorithms which biologists might use. And you can see that why they might struggle with this. I see no clear clusters here. Right, so the method that I would like to convince you is the method to use here is called k-cluster, and it is a graph-based clustering method. And it was presented by Bobrowski and Škraba in 2023. And it appeared in the context of their study of universality of distance-based persistent homology. Now, I won't go too much into their work on the universality, but maybe the main takeaway of it is that if you replace the additive measure of persistence by the multiplicative one, the persistence diagrams display some sort of universal behavior. So this multiplicative measure that they suggest is basically that you take, for each point, death divided by birth as a measure of persistence. So when we are doing clustering, this is basically zero-dimensional homology. So, and most filtrations that we tend to use have zero birth times for zero homology. So this multiplicative measure would actually not be the best to use. Actually, not the best to use. It's not valid to use it at all. So something that they were trying to do was think of another filtration that would basically eliminate this issue. Yeah, so I would just like to point out these two sources for this part. Right, so how do we define this filtration? As I said, we care about clustering, so we will only look at a graph. So let's start with a point cloud and build a complete graph on it, and then define the Vittori-Rips filtration on it as well. Now, as I said, we won't be working with Vittori-Rips filtration, but we needed to define the K-cluster filtration. So yeah, K-cluster filtration function will be defined on the graph. We need to fix a K in advance, which will be bigger than 1. And I will discuss the choice of K later on. But once we fix a K, we can define the filtration function on vertices as the first time at which the connected component of V in the Vittori-Rips filtration is bigger or equal than K. So basically you enter a vertex as soon as its connected component in the Vittori-Rips is big enough. Now on the edges, we take the maximum of the filtration values on each vertex and the distance between them. So basically an edge enters the filtration as soon as the connected components of both of the edges are big enough, and as soon as the two vertices are also close enough. So having defined the filtration function, this induces a filtration on the graph. And once we have the filtration on the graph, nothing is stopping us from applying persistent homology, which is what we love to do, and obtain a persistence diagram. Yeah. So let's maybe discuss this choice of K. So this is the most important parameter that we need to fit when we're using this method, but it is quite intuitive, and it decides the scale at which we consider meaningful things to happen. So to be precise, birth corresponds to merging two components of Vittori-Rips that contain, each of them contain less than K points, but when you merge them, they contain more than K points together. And death basically corresponds to an edge that merges two components that are both bigger than K in size, in a sense, killing one of them. Okay, so how do we go from having this K-cluster filtration and then the persistence diagram to clustering? So the first thing that we need to do is determine how many clusters we want to obtain. This might be motivated by your analysis. Maybe you know how many clusters you're looking for, but maybe even better, what you can do is look at the persistence diagram, compute the multiplicative value for each of the points, order them decreasingly, and then look at the gaps that you have in this sequence. And these gaps might give you a hint at how many clusters are inherently present in your data. So once you do that, you need to identify the threshold for the multiplicative value that separates out the n most persistent points. And once you do that, you can obtain the clusters following the tomato algorithm. So this is a agglomerative algorithm that merges things. And basically it merges things respecting your filtration, but here we also need to tell it that if the multiplicative value of the merged thing would be bigger than the threshold, then don't merge. And basically that's how we obtain the clusters. Now, let's see how it performs on our data set. So here we have both of the plots are with K cluster, where K is 8 and N is 7. And on the left, I use the Euclidean distance, and on the right, the cosine distance. And you see that they do quite a good job. So serotonin is nicely identified as one cluster. Octopamine, again, nicely clustered together. Tyramine is in 3 clusters, but we don't mind that. And also dopamine is in 2 clusters. So this looks quite good. And the cosine distance also looks good. Maybe the one thing I would point out is this area here, where we see by the representatives that there is a bit of a mess happening, and there's also some messy things happening with the clustering that we obtained, which might be a very good thing. But because I have no reason for preferring one to the other at this stage, I do the next step with the Euclidean, with this one, with the clustering for the Euclidean distance. So here I would like to then go on to task 2, where I just look at dopaminergic neurons. So what we need to do is get this cluster 2, which is green, and cluster 6, that is pink. We need to basically restrict our data set to those neurons only. And now we need to expand the analysis on all the genes again, because the 7 that we had for the previous task are no longer relevant. But we do some feature selection here, where we removed the genes that are whose expression is too uniform on these clusters. And what we end up with is this matrix DM, which has 458 neurons and 976 genes. So I would like to point out this task now is completely unsupervised. We don't even have representatives anymore. And we expect 20 to 30 clusters. So 20 to 30 clusters from 500 points, very unrealistic. And you will see that I did not manage. But yeah, I would like to show you my attempts. So firstly, I used K-cluster on DM, but I applied the square root for basically tackling scale issues. And the parameters were K equals 15 and N is 6 with the Euclidean distance. And what we see is that we obtain 6 nice looking clusters. They seem to be kind of looking nice on the plot. They're separated from the rest. Yeah, but there's really nothing more to say about this. So let's just go on to clustering 2. Here I used again the DM, but now without the scale, without the square root, and I used cosine distance this time. K was 8, N was 2, and I obtained 2 clusters. I would like to point out that cosine distance really struggled with clustering these further, so I just stayed with this. And then clustering 3 was, I basically performed a way stricter feature selection. So if I had around how much was it? Around 1,000 genes here. I had around 120 here. So I really was quite harsh with the feature selection. And I again plotted this. I used Euclidean distance. I applied square root, and the parameters were k equals 10 and n equals 4. And I obtained these clusters. Now, none of these clusters are impressive on its own. They're just some pretty plots. But maybe what sells it, at least to me, is how they look when I plot them on each other. So the points here are plotted as they are for clustering 1. Also, the colors are the same for clustering 1. But the annotations, so the shapes of the points that I used, are from different clusterings. So here it's from clustering 2. And you can see that the cluster 1 is basically here, and everything else is cluster 0. And I mean, it doesn't agree with the clustering per se, but we like that it is kind of separated from the rest in the plotting. Then clustering 3 on clustering 1 is maybe even more, it's selling this even more, in a sense that we have cluster 3 being all these big clusters together, but then it agrees exactly with the clustering on these 3 clusters. So these 2 clusterings agree. And then just for the sake of having all the plots, this is the clustering 2 on clustering 3. And we see that also on this plot, this cluster is kind of separated from the rest. Right. So as far as my data analysis goes, this is where I stopped. I am currently waiting because my collaborators are expecting to have a new data set with many more dopaminergic neurons very soon. So I hope to continue this. I hope this to be kind of a proof of concept for future work. And I hope I will be able to update you on it soon. But maybe let me also say that I think the main selling point for K-cluster method is its ease of use. And here there are many points that I would like to make, namely, it is very applicable in a sense that you can choose whatever distance you want. You can choose different things to do to your data, and you don't have to have specific assumptions about the distribution of your data for this to for this method to work well. And secondly, the only real parameter that you need to fit is this k, but this k comes with some sort of intuition in terms of the scale of important things happening. So I actually, when I was doing these sort of analysis, didn't spend a lot of time fitting parameters. And lastly, the amount of clusters that you can obtain, you can see from the persistence diagram. So I think that is also another selling point for this. And to end the talk, I would also like to make some I would like to thank some people. So first of all, Škraba and Bobrowski for making an implementation of K-cluster available to me. Then again, Vincent for sharing the Thirsty Fly dataset with me. Biophysical Sciences Institute at Durham University for fostering this collaboration, and of course AATRN for inviting me. And just some sources for whoever wants to look into this further.
All right, thank you very much. Let's all briefly unmute and clap for Živa. All right, thank you so much. So we already have actually one question in the chat, which I'm happy to read out loud. So Daniel Tolosa was asking, why do you expect 20 to 30 clusters?
So this is this is basically the information that was given to me by my neuroscientist collaborators. They kind of expect there to be 20 to 30 cell types. But that's all that I can say about this. This was information given to me by my collaborators.
Okay, interesting. I mean, Daniel, if you're still around, please feel free to unmute, of course, and then ask follow-ups. We have another question, I think, from Marcus. Please.
Hey, great talk. I have an oddly specific question.
Okay.
You said you used the square root to deal with scale issues, or one of the attempts to use.
Here.
So I know that in the single cell community, often people use the logarithm to deal with as a variance stabilizing method. So what's your take on square root versus taking the logarithm?
To be honest, I think both of them are completely valid. I just tried to avoid the logarithm because the data has a lot of zeros, and then you need to basically do this dance of moving it and then moving it back or something like that, which I didn't want to do. And square root seemed to perform equivalently well. So I think either way is fine. Whatever you use is fine, I think.
Right. I think next we have a question from Dave. The question is a little bit general. So if your audio works, oh yes, please go ahead. Yeah.
Yes, I have. I'd like to ask about two methods, which I don't believe you mentioned.
Mm-hmm.
One, which is often used in single cell data analysis, is UMAP, but that's often used. But perhaps that's used more for larger gene sets, thousands of them. So I don't. If you had thought about that,
So UMAP is used for dimensionality reduction. So it is basically when you have a very large data set and you want to, in a sense, it is in a lot of dimensions, and you want to first project it into a lower dimensional space so that your other methods that you're trying to use work. So it's not necessarily used for clustering, but kind of as a companion to clustering. But I did use it for the plots. So in order to plot the data, I needed to project it to 2 dimensions, and there I did use UMAP, yeah.
Right. So then when you use UMAP, you can also tune UMAP so the plot has different characteristics. So for a large number of nearest neighbors, it's more spherical. Smaller number, it fragments. So you were somewhere in between then, I gather.
Yeah, to be honest, I didn't spend a lot of time tuning the parameters of UMAP because I just needed it for the visualization. But there are, I think, plenty of things that you can tune there. But here I'm not the expert, so I would, I would rather not say anything to lead you astray.
Sure. I have a second question then about a different method, which is Mapper.
Yes.
If you've tried that, I know Rabadon applied that to single cell data.
Yes, he did. I think I know exactly the application you have in mind. I think the setting of that one was slightly different. Correct me if I'm wrong, but I think he was studying the differentiation of cells. So he started with these, how are they called? I know it in my language, I don't know it in English. So he basically started with these cells that are in the bone marrow. How are they called? Basically the cell before it differentiates into its type.
Right.
So he started with those and then tracked the differentiation into different types. So the difference there is that you have some sort of continuity in your data.
Right.
When I tried to apply it to this data set, it didn't work that well. And I think it might be due to the fact that there is no continuity like this that would connect things. And also this data set is very prone to outliers. And I had plenty of them here. And this is something that I struggled with when I tried to apply Mapper.
Yes, Mapper can give you many outliers and they just float around outside the graph. Yes, good.
Yeah. Yeah.
That's good. Thank you.
Thank you for the question.
Welcome.
Very exciting. Any additional questions on this? Maybe I can continue with an overarching one. So what would you say in summary are the main properties that make persistent homology or TDA in general, I should maybe say, advantageous or interesting in this context? Because I always find it hard to. I find it amazing that this worked and that you have nice collaborators. I find it very hard to explain and to talk to my collaborators about these things. So I'm trying. Maybe I can get some tips here.
Yeah, that's a very good question, and I should really think about this before the talk as well. But I would say that maybe persistent homology is very flexible in a sense that you need to set up your analysis in a right way, and then it gives you good answers, whereas a lot of other methods make a lot of assumptions that because of those assumptions, you're not allowed to manipulate your data in a way that you can in order to use the persistent homology. I don't know if that's a good explanation, but that's kind of how I
think that's good. Mm-hmm.
Yeah.
Very, very nice. And maybe a follow-up on this, unless there are there other raised hands that you briefly check. Sorry about that. And in terms of the computational complexity, is this what are we talking about here? Is this feasible? Because it seems like a very nice pipeline that you're presenting here.
Yeah. That's a good question. I think K-cluster is not very expensive, especially not compared to other graph-based methods, but I don't know the details. For the analysis that I've done so far, I worked with this and another data set, and for this one, it was very easy to use. It also computed things very fast, and actually, usually K-cluster was done in seconds, whereas the visualization might have taken long. And this is true also for another data set that I've been working with, which is a lot bigger. But yeah, here I actually don't have a good answer. Apologies.
No, it's all good. I mean, this is a genuine interest, right? I mean, it's just figuring out where this might go. And okay, I mean, I can still continue. I mean, if people are still around and no one has direct follow-ups. So one thing that I find very interesting about this scRNA-seq in general is how the data looks in a way, right? So can you remind me, or maybe the others, maybe someone else forgot, how your input data looks in general? Is it essentially, do we have to think about this as matrices of genes and expression levels and things like this, or samples and expression levels?
Yes. The way I think about it is basically a matrix. And I think of cells as rows and columns being the genes, but you can also, transpose it. And each entry is kind of a count, so it's an integer of how many times a specific gene has been copied in the gene expression of that specific cell. And yeah, maybe one thing that I didn't quite point out enough maybe is that this is a very sparse matrix. It has plenty of zeros in it. Yeah, and it is very prone to outliers because every now and then you just have an expression that is extremely high. So yeah, it's quite interesting to work with.
Yeah, maybe this also answers one of my takes, namely, I think persistence is great here because it gives you a way to choose different ways of working with the data, different similarity metrics, different distance measures, and so on, right? And so you could kind of still get those results. Oh, that's really great. Thank you so much. So are there any further questions from the audience? Don't be shy. Okay, we have another one. Please go ahead.
Yes, you mentioned just your integer values. Often with single cell data, you need to normalize it in some way. So does this mean that there was no normalization that was going on?
No, other than the times when I used the square root. I mean, that's not normalization, but it was trying to take care of scale differences a bit. And then the cosine distance, the cosine similarity is scale invariant, or not invariant, but yeah, invariant. So basically scale doesn't affect it. So whenever I was working with cosine similarity, this was kind of actually ingrained in it. But that's basically, I think that's scaling for each neuron, its value. So I could have done similar for each gene, to scale each gene. And I've tried many things here, but nothing performed close to as good as these things. Or, I mean, it was unnecessary to do it because I didn't gain anything. Yeah.
Good. Thank you.
Right, great. Any other questions from the audience in the recorded part? Because if not, I'm going to stop the recording and we can have some offline exchange. So again, thank you very much for a very exciting talk on scRNA-seq and TDA.
Thank you.